Bismillahirrahmanirrahim. Assalamu alaikum. Hello and welcome to Biosciences with ShahTareen Swati, and I'm here to deliver the lecture about the tissue culture today. And I'm not only going to deliver the lecture, but I will also provide the stuff for your notes because mostly students ask me to provide the stuff for their notes. So here not only listen to the lecture, you hold on your notebook, pen and pencil, and make your own notes from my lecture. And in this lecture I'm going to discuss about what is the tissue culture, who discovered the tissue culture, what is MS medium and its discovery, equipment we need for the tissue culture, and different steps of the tissue culture in detail. So first of all, what is tissue culture? Tissue culture is the cultivation of the plant cell, tissues, or organ on especially formulated nutrient media. Under the right condition, an entire plant can be regenerated from a single plant, sorry, single cell. So, in simple words, tissue culture is what? The growth and development. This is the artificial growth and development of the plant cells, tissues, or organ in a specific environment, such as in a lab, to get the whole plant from the single cell. That plant must be disease free, that must be high quality planting material, and the reproduction we need high yield, so we can produce by using the tissue culture. Plant tissue culture is a technique that has been around for more than 30 years. Tissue culture is seen as an important technology for developing countries for the production of the disease-free, high-quality planting material and the rapid production of many uniform plants. Is it clear? Okay. Now, who discovered the tissue culture? Because mostly students don't know who discovered the tissue culture and how it was developed. So, William Rocks in 1885 removed a section of the medullary plate of an embryonic chicken and maintained in a warm saline solution for several days, establishing the basic principle of the tissue culture. And the second one, Ross Granville Harrison, an American zoologist, was able to culture the nerve cells from a frog in solidified lymph. Because of his contribution to the tissue culture method, Harrison now has the title of the father. He successfully grown artificial tissue culture. So the difference between the experiment of the Ross and Rocks is that Rocks put the chicken embryonic medullary plate into the saline solution, and Ross used the solidified lymph to grow the nerve cells from the frog. And you can see the photograph of the Ross and the rocks on the right side of the slide. Okay. Now, what is Murashige and skoogmedium, which is commonly known as, and you people are familiar with, MS medium. Maraschi and skoogmedium is a plant growth medium used in the laboratories for the cultivation of the plant cell culture. MS was first invented by the plant scientists Murashige and Fouke Skoog in 1962 during Murashige's search for a new plant growth regulator. A number behind the letter MS is used to indicate the sucrose concentration of the medium. For example, MS0 or MS0 contain no sucrose, MS20 contain 20 grams of sucrose. Along with the modification, it is the most commonly used medium in plant tissue culture experiment in the laboratory. Okay. On left side of the slide, you can see the Murashige, and on the right side, Skoog. I have the photograph of both because I didn't see Murashige before, but when I was searching the stuff for the tissue culture, I found this picture of Murashige, and now I'm sharing with you people. You must go through the Murashige and Skoog life history. I'm sharing all the scientists with you people to introduce these inspiring people, because they spend their life for the science and technology. Actually, they spend their life for the humanity. So, must pay tribute to all these great persons. Okay, now equipment. I'm going to share what equipment we need for the tissue culture. We need the tissue culture hood, tissue culture incubator, laminar flow hood, inverted microscope, dissecting microscope, pipettes, standard tissue culture equipments. We need the centrifuge, freezers, refrigerators, and water bath. And most importantly, we need the autoclave for sterilization purposes. So the steps of the tissue culture. First of all, the selection and sterilize. Sterilization is the basic step, which is very important for the tissue culturing. If you want to do the tissue culture in the lab, you just sterilize all the equipments, explant, each and everything which you are going to use in the tissue culture. Is it clear? So first is the selection. Select a healthy parent plant. If you want to get the mass of the cells, the tissues, or any organ from the plant, that is known as the explant. So eliminate any microbial contamination from any exposed explant surface. Now, cut the plant into small pieces, note down all the points, and using detergent, second point, and water, and then transfer the plant part into a sterilizing cloth solution, then using the lid, and then save, as you can see here in the figure. Okay, now the medium preparation. The appropriate mixture, like the MS mixture, is mixed with the distilled water. So must keep it in your distilled water, even stirrer, each and everything must be sterilized. So first, what will happen? The appropriate mixture, like MS medium, mix with the distilled water and stir while adding the appropriate amount of the sugar, like the sucrose. Here, sodium hydroxide or hydrochloric acid is used to adjust the pH, and agar is added to the mixture. Heat and stir to dissolve, and after cooling, the warm medium is poured into the polycarbonate tubes. Okay, now the next step is inoculation. And you can see here, this is the laminar flow hood, and all the steps of the tissue culture must be performed in this laminar flow hood. Placement of the explant in an adequate culture medium. Inoculation of the explant is done in a controlled environment with appropriate temperature, humidity, air quality, and elimination done in a laminar flow. Now the development of the callus. What is callus? Callus is basically the mass of undifferentiated cell. In this technique, plant cell propagate on suitable medium to form clumps of the undifferentiated cells called callus. As you can see here, the development of the callus in the plant. Callus contain dedifferentiated cells. Changes to most must take place before cell division can occur. Balance of oxygen and cytokinin help in cell and callus development. Development of the plant. After callus formation, callus is then shifted to the new media for the development of the shoots and roots. The mature plantlets can be transferred to the plotting soil for further growth in greenhouses as normal plant. Okay, thank you very much, and I will be back till then. Allah Hafiz.